Have you ever wondered how a tiny piece of a plant can grow into an entirely new organism, without soil, sun, or even seeds? Welcome to Biotic World. Today, we're diving into the fascinating history of plant tissue culture, a technique that revolutionized plant science, agriculture, and biotechnology. The idea of regenerating plant tissue isn't as modern as you might think. It all began in the 19th century, when scientists were still discovering the very basics of cells and plant physiology. In 1838, Matthias Schleiden and Theodor Schwann proposed the cell theory, stating that the cell is the basic unit of life, a concept that laid the foundation for plant tissue culture. The earliest practical attempt to culture plant cells was made by Gottlieb Haberlandt, an Austrian botanist, in 1902. Often called the father of plant tissue culture, Haberlandt proposed that individual plant cells could grow independently under the right conditions. Although his experiments didn't succeed in producing a whole plant, his visionary work led to a new scientific field. Between the 1930s and 1960s, plant tissue culture evolved rapidly. In 1934, Philip R. White successfully maintained tomato root cultures in vitro. Around the same time, Gauthier, Nobecourt, and White developed continuous callus cultures from carrot and tobacco, a major milestone. In 1955, Skoog and Miller discovered that the balance between auxins and cytokinins controls shoot and root development in tissue cultures. By the 1970s, plant tissue culture wasn't just a lab trick, it was a full-blown industry. With the technique of micropropagation, scientists could mass-produce identical, disease-free plants from a single cell. This transformed crops like bananas, orchids, sugarcane, and potatoes. Fun fact: over 500 million banana plants are grown every year using tissue culture methods. Today, tissue culture is a foundation for plant genetic engineering, germplasm conservation, CRISPR gene editing, and even synthetic seed production. It allows researchers to create transgenic plants, preserve rare species, and develop climate-resilient crops, all starting from a tiny explant. So, from Haberlandt's glass slides to genetically engineered supercrops, plant tissue culture has come a long way, and its journey is still unfolding. If you found this fascinating, hit that like button, subscribe to Biotic World, and tell us in the comments which tissue culture discovery surprised you the most. This is Sheetal, bringing science to life. See you in the next video.